The nucleus is a structure that we find in pretty much all eukaryotic cells, and this is really where the name eukaryote comes from. This, a lot of times we like to think of it as being the brain of the cell, and the reason we're calling it the brain of the cell is because this is really responsible for controlling the cell's activities. And it controls the cell's activities because it contains the majority of the DNA that you have inside of a eukaryotic cell. So it does contain most of the DNA, and that DNA that we do find here is going to be in the form of linear chromosomes. And there will be quite a few of these linear chromosomes. So if we talk about the overall structure of the nucleus is going to be a membrane-bound organelle. And so we do have a membrane around the outside. And if we label it here, this is the membrane, and this is what we call the nuclear envelope. And this nuclear envelope is actually a double membrane. So there's kind of two phospholipid bilayers there. And the nuclear envelope is going to be responsible for allowing entry and exit into the nucleus. And this is important because the nucleus has the DNA inside of it, which is the blueprint for everything. So certainly there is going to be a lot of entry and exit from the nucleus, and it's going to occur through that envelope. Because we have all of this entry and exit, there does have to be pores there in the envelope to allow molecules to get in and out. So I'm just going to go around and kind of draw them like this to show little holes in the envelope. But these are regulated holes, meaning things aren't just leaking in and out. So these little holes that I'm drawing, these are what we refer to as nuclear pores. Okay, so if we label those, the nuclear pore is just part of the nuclear envelope, and this would be the structure that molecules are actually passing directly through when they're entering the nucleus. And this would be to get information out of the DNA. So if we need to read some information in the DNA, then we would have to pass through the nuclear pore. If we look at where the DNA is at in all of this, the DNA is going to really look like it's not very organized inside the nucleus, because most of the time it's really going to look kind of along these lines if we were able to see it, but we wouldn't most of the times really even be able to see it. So this is my DNA. Again, that's in the form of a bunch of linear chromosomes. And while it looks like a jumbled up mess, the way that I've drawn it in there, it's not a mess at all. It's very organized. And the way that it's organized is there's going to be a network of fibers just on the inside of the nuclear envelope. And so I'm drawing some of that in right here. And so that would go all the way around. This is going to be what we refer to as the nuclear lamina. And the role of the nuclear lamina is to organize the DNA. And this is important because there's a lot of DNA there, and that DNA contains a lot of genes. So we're really talking about thousands of genes. And so the nuclear lamina is going to provide attachment points for the chromosome. So in other words, you might have some of the DNA attached right here, kind of tethered onto the nuclear lamina, some up here, some over here. And that's going to help the cell to know exactly where each of the genes are located, or in other words, where they're going to find the information for making a particular protein that they might need to make. So we're going to find that very often, really every second of every day, that we're going to have different molecules entering through the nuclear pores and the nuclear envelope, going in there, finding particular DNA sequences and looking at that information, because it is the information to make a lot of the proteins inside the cytoplasm or out there bound to the endoplasmic reticulum. Also inside the nucleus, we do tend to a lot of times see some really dark staining spots. And so if we draw that in here, there might be a spot over here that's darkly staining. There might be a couple of them. So it's usually a small number, maybe 2 or 3 that we happen to see. And these structures are what we refer to as the nucleolus. And so if I label that here, the nucleolus itself does not have a membrane around the outside, so it's not an isolated compartment inside the nucleus at all. But it does tend to stain a little bit darker when we stain the nucleus. So this tells us that there are some different molecules found there compared to the rest of the nucleus. What we tend to find there is we find lots of ribosomal RNA, or what we call rRNA. So this is really where that rRNA is going to be produced, and rRNA is a component of the ribosomes. So the nucleolus, we can think of it as the site of ribosome assembly, or in other words, where the ribosomes are actually put together. And just because of those components being present there, it does cause that area to stain a little bit darker than the rest of the nucleus. Okay, so that's a look at some of the general features of the nucleus. The nucleus is going to contain a bunch of linear chromosomes, which makes up most of the DNA that we do find inside eukaryotic cells.